Okay, so Corey and I took quite a few evolution classes together, especially evolutionary theory, and we spent a lot of time talking about it. And what I invited him here to do right this second, which he does not know yet, is I wanted to kind of brainstorm with him about how you're going to synthesize your evolutionary interviews. So you know this, that everybody's picked an evolutionary biologist. They have interviewed their evolutionary biologist, meaning they read papers that person wrote or that's been written about that person, and answered questions how they feel like that person would answer them. Okay. These questions are things like, what are the primary mechanisms that are driving evolution? Whether or not if you started completely over, you would get to the same point. That's the classic
the
Carol. Yeah,
tape
is that
of life.
Carol that rewind? That did
I
the
think
rewinding?
that's actually Gould.
I thought it was Gould talking about Carol.
I don't know.
Anyway, these type of questions. So along with like basic, this is what I study, this is how I fell into being an evolutionary biologist, because nobody starts really by being an evolutionary biologist. And so what I want them to do next is synthesize this, because we don't have time to go through like step by step Darwin and Wallace, Lamarck, Cuvier, Mendel and the new synthesis and all of that. So I want to kind of put them in clusters or something so that they can go like this biologist versus this biologist. And so I wanted to kind of get your, like, why is that important? Why should we be comparing people's different evolutionary theories?
Right, because there's some classic
disputes, would you go?
I don't know if disputes is right, but there's definitely some classic debates that led to the fleshing out of the parts of theory that are interesting. So, there were classic fights between mutationists and selectionists. And in the process of having that argument, each side sort of collected data. And in the process, they actually gelled to something that's close to the biology we know today. And all of these different debates have ended up being about moving the entire field forward.
Right.
And so I think they're actually really crucial. If you can see that this biologist is coming to an idea from a neutral theory perspective, and this person is an adaptationist, it's not these two camps that can never join forces. A lot of times what comes out of those is a better understanding of the entire structure of it.
Right, and I think that's kind of the thing I want to get at is when you're learning about various people's perspective in evolutionary biology, it's not that they think the other perspective is wrong. It's that this is the thing that they think is most interesting and or most important about evolutionary biology. Now there are you got some major leaps that you're like, oh, that person was wrong, for instance.
So I think a for instance on that would be selfish gene theory. Selfish gene generally doesn't hold up very well.
Give him a little brief rundown of selfish gene.
Which is Dawkins' idea. And so selfish genes is the idea that your genome is the activity of a bunch of individual selfish components, and that those particular components are acting on their own, and that your body is basically just
a sack where they're all interacting.
A sack to get genes to multiply as many as possible. It's problematic for a lot of reasons. It puts the idea of motivation on a biochemical reaction, which is unlikely to be the situation. It also ignores the complete reversal of the idea, which is that it's in fact the body that uses genes, and that genes are more plastic. It also ignores a general holistic idea that genes interact and genes genes, that the more important part of your genetic system is actually the interaction of those genes, not any individual component in most cases.
And I think what is a major problem is, which is a common thread, is that people define, what they think is the most important part or leading mechanism or whatever, and they have this, even without trying, they have this motivation. Like, there is a motive here. And there's never a motive because we're drift people.
Right.
So, it's always random.
And so I think the benefit of that whole way of looking at evolutionary biology has been to see where it fails and where parts of it can hold up. And in fact, what I was talking about earlier with bacteriophages or something like that actually may be a really good place for a selfish gene. You have a part that really doesn't care much about the organism. Sometimes they'll impart a lot of really cool things into the organism's genome, but it is just to get a free ride. Viruses probably live that way. Eukaryotes probably don't.
Another thing that I think is important is that a lot of people use this discrepancy between evolutionary biologists as a aha, this is why evolution can't be true. But that's not. If you talk to any of those evolutionary biologists, whether it's in fake interview or in real life, that's not the point.
I think it would be wrong to look at these debates as a wedge to drive a big hole,
right,
or to knock down evolutionary biologists. These are conversations that, in the right form, are debates among specialists.
Right.
The same thing happens in most fields of science. Up until very recently, it was a big debate about the existence of dark matter or black holes and stuff like that.
That's another good point.
And it was through scientists asking other scientists to prove it.
Yeah.
Let's not move too far in the area of criticism. So there is the point where you have to be critical, and anyone who's going to posit an idea that has broad implications has to have an extensive set of data, and you can define what that data would be. But simply saying, all right, because of that, we're now going to deny the mechanism of antibiotic resistance, we're going to deny the mechanism for biodiversity of the planet, we're going to deny the mechanisms that all of biology is currently based on, because one of these debates, that's definitely the wrong way to go.
Another thing I think you should keep in mind, and a lot of you are doing current evolutionary biologists, which is cool, and it's going to be interesting for me because I don't know that there's some on the list that I'm going to have to read up on. But also you have to remember that there was a time when everything wasn't just happening in the open. Do we argue that everything's happening in open right now? No, because they're under paywalls and everything like that. But where literally people would mail people on ships documents for them to read about their ideas. And this was a very slow debate. That's how you could have people with very drastically different ideas that felt like they were drastically different. Mostly because they weren't sharing information fast enough to be like, oh, I get it. This is how this relates to my thing, and this is how it relates to your thing. It was just I don't know, 100 sentences back and forth every 3 months.
Well, you can imagine Darwin in his Victorian home receiving word from Huxley that he'd heard from Wallace that he was about to completely usurp his idea, and it lit a fire under him for sure.
So you got to write faster
to publish, and it's the same way.
Get a pigeon.
It's the same way now. I mean,
yeah.
It definitely there's sayings in evolution that the closer two species are, the more competition actually exists, and that your real competition is actually not generally interspecies but within species, and the closer any two groups are, the stronger the fight is.
Okay, one more thing, because nobody picked Lamarck.
Uh-huh.
So we get to kind of do the classic Lamarck thing. Let's give us a brief rundown of Lamarck, because I know you know it.
Yeah, so Lamarck is all about the acquiring of heritability.
Acquiring of heritability and descent. What's it called? Descent. Oh, gosh. Up the ladder. What's that thing called?
The chain of being. Chain of being.
The great chain of being. So
the idea is that organisms start off as rocks and move all the way to angels.
There's like a clip art.
Yeah.
I will show you.
And so we have this point where we have rocks, and the rocks would really do well if they were sort of slugs.
If they could move, because
if the rocks
somebody
could
sat
move.
on them or whatever.
So the classic idea with this is a giraffe is trying to get the tallest part of the tree to eat, and so it just stretches its neck and it keeps on stretching it, and in the process of stretching its neck and getting a stretched out neck, it ends up over time having a longer neck through evolution.
Because the mom's stretching her neck and the dad's stretching their neck to reach the tops of the trees, and then so their child's going to have a longer neck, like as in it's additive.
So
some way the inheritance is additive of the phenotypes. So then those two, that the offspring have longer necks, but they're still stretching their neck to reach the top of the trees, and then there we go, and then we have a giraffe. What's the major problem with that?
Well, the major problem is we see nothing about that in the actual record of biology. There's no...
As in, giraffes don't actually eat from the tops of trees. The giraffes just eat whatever. It's like a goat.
Yeah.
With a long neck.
Also, you don't typically see people who've lost arms in boating accidents having children with that same missing arm.
Right. Which was the classic... It's all in the wording, obviously, but words are important. So that doesn't work out for Lamarck.
Yeah, so it doesn't hold up even a little bit. But I will be interested to know what your students read about the current state of epigenetics.
Yeah. Let's go back there in a second. Let's finish our giraffe concept. Turns out giraffes have long necks because...
of sexual selection.
Ooh. Here's the thing I don't like. The difference in our camp right here is that I think sexual selection is just selection. It's just selection. It doesn't matter.
It is in mate choice.
Mate choice.
So what happens is there's this period of fighting between male giraffes.
For territory.
For brood.
But the broods live in a place. So...
Right.
I get huffy about sexual selection.
So they fight each other for control of that territory.
With their heads.
With their heads and necks.
And if you have, it's torque.
Yeah, big, long, swinging neck.
The longer your neck, the more you can swing, the more momentum you can build and whap. And you should now YouTube that because it is terrifying. It makes the loudest noise, even just on a video.
And so what they found out was they were studying on trees the height at which they actually ate. And it turned out that most giraffes were eating from a position on the trees that was actually more uncomfortable because of their long necks.
And have you seen them drink out water? That's just not really rational.
It's also unlikely to be a survival type benefit because when you think about if there's going to be a situation where the entire bottom of a tree is picked from, but the top isn't picked from, it's the rarer of situations. And the interesting thing about selection is that you just have to barely make it.
Right, and they've done that in the E. coli migration. the megaplate.
Yeah, you do not have to...
You see, like, it's not survival necessarily of the fittest. It's just fit enough to get to the next zone. And that's what they're doing with giraffes. So the male giraffes that get the biggest territories may have these dumb, giant head-neck ratios, but they are mating. And that's all that matters. So there we go. Let's go back to, so that's the end. Mark was wrong. Bummer. Giraffes was a bad choice in his for example because it is maybe the most obvious, like that took like a week of just observation to prove that wrong.
Yeah.
Let's go back to this, what our students are reading about epigenetics lately, and I will caveat this with, I was talking to a friend of mine who's like, I don't know, what a person would call a science nerd. So not a scientist, he's like a high school teacher in English, but he listens to science podcasts, and he said, what are you going to do about evolutionary theory and epigenetics? It's just ruining everything. And I was just like, I don't know what we're talking about because I don't keep up with the science nerd culture, like the podcasts about science. And so here, I think this is where you're going to fill it in.
Yeah, so there are a couple mechanisms.
We should define epigenetics.
Yeah, so it is genetics. It is a heritable trait usually that happens outside of the primary genome.
Has to do with, or we think it has to do with, how the DNA winds and unwinds. So for expression to happen. So it's about expression.
Right.
Not sequence.
So the sequence doesn't really change ever.
Right.
You have your sequence. However, within your lifetime, you can acquire certain modifications of your genome. For instance, you can acquire what are called CPG islands, which is an area that silences or turns off a particular cytosine.
What does CPG stand for?
It's literally CPG. It's just a phosphate between a cytosine and a guanine.
Mm-hmm. Okay.
And so there are these little methylated areas that latch on to cytosines, and what ends up happening is it silences a particular point within the genome through this methylation effect. So this methylation sits on top.
Those are the tags that influence unwinding.
And so it turns out that, for instance, mothers who smoke may acquire a large number of methylation sites on cytosines, which causes them to be particularly silenced.
Another one is trauma. Trauma during pregnancy can often, or is thought to, cause a lot of these weird methylation patterns that you don't see in other cases.
So you may carry the same genome, but you may have in your lifetime, through certain behaviors or certain incidents, acquired parts of the genome that are then transferred to your offspring. And the result of that is a change in phenotype that you acquire over your lifetime. So not a change in phenotype that is related to your
without a change in
to
phenotype.
your sex chromosomes.
So it's a change in phenotype without a change in genotype.
Exactly. Without a classic change in the genetics.
I think you could argue that that's genotype.
I don't know. I think you probably could. Yeah.
Just to go back, I'm sure you guys have read this example because I'm pretty sure it's in your genetics book. This is why they think you can have identical twins and one can acquire cancer and one cannot. It's largely, and also why identical twins start looking more different as they age. It's because they're acquiring these methyl sites, these epigenetic markers that are changing gene expression.
So the most interesting studies in human genetics are the twin studies.
Yeah, let's go back to why this is why this relates to synthesizing evolutionary theories between biologists. How is this changing evolutionary theory?
Well, it's explaining some of the variation that we see. I would say that it's not critically affecting our understanding of biology. The classic biological mechanisms still hold as much as they ever did.
Right.
The issue is that there was always a bit of variation that we didn't understand where it came from. And a lot of times those little bits of variation end up inspiring a whole field within biology or within science in general. We'll see some part of science that can't be explained and is sort of lost in the random element in the noise
What used to be
element.
called like junk DNA or noise, and it starts being like, oh, those repeatable elements are really important. Oh, these weird palindrome areas are actually really important. That was my answer to this person, my science nerd friend, was this just another explanation of variation or another chunk of the variation between organisms explained. And like, maybe it's because I am more of a drift person than a selection person. Like, I'm okay with that.
Yeah.
Because obviously,
So
like, there's going to be.
I think if you.
I don't think it like breaks any rules.
I think if you're a person who thinks that selection is this highly tuned machine and that organisms are highly tuned by selection to be these, powerful racehorses or powerful examples of everything in your history, just working under the constant scrutiny of selection, I think it may be a bigger pill to swallow.
Yeah.
But I think a common, more nuanced view of the way that evolution works fits this mechanism just fine.
I mean, if you were in my 310 class, you know that you just always assume that the rules are broken. Like, oh, you have gram-positive bacteria and gram-negative bacteria, and those bacteria that can't be gram-stained. Gram-stained is both, whatever. So you always have things that break the rules, and that's because evolution is, happening. I thought of something while you're saying, when you're talking about these people that are really like fine-tuned racehorse selectionists, who are you talking about?
Oh, I don't even know if that many currently.
Let's give it a prior example.
A prior example would be Gillespie, who was the person who really attacked Kimura's neutral theory. So there was this period of time in the 70s when Kimura and Crow and Oda put this idea of a molecular clock, and that there were just these periods of selection. Really, the entire modern synthesis. If you look at a lot of the modern synthesis people, Sewell Wright in the early stage, a lot of these people who really rejected that drift held the place that it did, or that really random events in general held the place, but who believe that there's this whole selectionist landscape and everything is at sort of its peak.
Yeah.
I think for any of those people, this would be a tough pill. But for someone like Michael Lynch, who is completely on the other side, who thinks that selection has almost no,
Right.
I would say, is probably even an advocate on the other side, on the neutral side, that most organisms are not under very strong selective constraints at all, that it's pretty easy to get by in the world for most organisms, and so most aspects of your genomic architecture, his words, are sort of floral.
I think one thing that we should point out is that Michael Lynch, one, ties in epigenetics really nicely because he did that old male sperm.
Right.
He is an old male. who had a child very old. He would say that.
That was what the talk he gave at University of Missouri was about.
But that old male sperm has collected enough of these epigenetic markers that they actually think that it's not advanced maternal age in women that can cause some major problems in birth defects and whatnot, but actually potentially the sperm.
He's one of those definite iconoclast National Academy members.
Yeah.
Who definitely ruffles a lot of feathers.
He upsets a lot of people. Another thing that we should point out is he might be, as cool as Megaplate is, he might have been the first Megaplate, because instead of looking at these like rapid mutation and phenotypic changes and by rapid I mean watching physically with your lifetime and your eyeballs, how evolution is occurring. And he did this in water fleas, which are a tiny crustacean. And they're easy to keep in the lab in aquariums, and so he just let them be. They all had food, they all had water, there was no like gradient of selective pressure like in the Megaplate, and then crazy things happen, and maybe I'll pop some of those in there, because I don't think any of you are doing Michael Lynch. Maybe you are. But
I think it's the value of really knowing your study organism, right, and just being willing to do the boring stuff to answer a really fundamental question in biology.
You'll see, like, Lynn Margulies was just doing SEM of different organelles. That's how she came up with her idea.
Hopi Hoekstra, and just looking at Peromyscus.
Yeah.
She had this organism that has been studied so well, Mus musculus, and then she looked at an organism that's pretty closely related, but it is a wild version.
Yeah.
And just said, how do they evolve this really complicated behavior?
And really, they started by just looking for a wild type version of the thing they were studying. So they just wanted, like, what does it look like in nature? Not what does it look like in this other species? Let's do an evolutionary study, like that kind of thing. Michael Lynch was an ecologist. His advisor was a famous ecologist looking at lake systems in like the Great Lakes area. But yeah, he was just measuring chemical differences in lakes and how you saw whether or not you saw different types of species. So,
and it was the same thing with Coyne, looking at wild Drosophila and clinal variation. We had this view of sympatric speciation. We had a view of speciation that happens when two organisms are really far apart, but what if you put them partially close together? And really studying that, some of these fundamental questions have been answered by people really just willing to do the experiment that takes forever, requires a lot of time, and
observation.
And just steady, solid observation.
Yeah, just like sit there and watch it. Okay, so that was kind of why we're going to do this synthesis part and how we're going to kind of do it. You guys are going to have a spreadsheet through Microsoft, because we are Microsoft school apparently. That's going to be the easiest way to do it. And you're going to compare and contrast your scientists. But, yeah, that's pretty much it. Thanks. Thanks, Corey.
Yep.